Greetings. My name is Curtis V. Smith, Professor of Biological Sciences at Kansas City, Kansas Community College. I'm going to do a presentation on coronaviruses. And this is for the Academic Symposium at Kansas City, Kansas Community College. The Center for Teaching Excellence there provides this opportunity for faculty. Our mission is to provide an open and public forum which will allow the faculty and staff of the college to make presentations of academic or artistic merit. As faculty and staff of this public institution, we seek to uphold the values expressed in the Academic Freedom Statement, which says that it is essential to the fulfillment of purposes of higher ed, and we acknowledge the fundamental need to protect faculty members from censorship or restraint which might interfere with their obligations in the performance of professional duties. Accordingly, faculty members shall be guaranteed full freedom in presentations and discussions and may introduce politically, religiously, or otherwise controversial materials relevant to course content. When faculty members are exercising their rights to citizenship in public, they should indicate they are not official spokespersons for the college, and I certainly am not. The views and opinions expressed during this event are those of the participants and do not reflect the official policy or position of the college. KCKCC is an equal opportunity and affirmative action educational institution. I want to thank the Center for Teaching Excellence at Kansas City, Kansas Community College for this opportunity to make this presentation. It's in essence the biology of the virus that I present in my courses, and I think everybody will have some level of appreciation for biology. Everybody comes to this with a different level of knowledge, and I want to present it basically at the community college level. I've been teaching microbiology for 26 years there at Kansas City, Kansas Community College, and this topic is something that is important for our pre-nursing students. We have a large pre-nursing program and a nursing college at KCKCC. So, but this is for general knowledge, general information. I think that the goal of this being a biology presentation is really to preclude any extremes or a false sense of security or fears either way, which can be harmful and have social consequences. So the more you know about the virus, the better off you'll be. My sources are the United States Center for Disease Control in Atlanta, and also I would recommend Methods in Molecular Biology from 2015 by Furr and Perlman, University of Iowa publication, very detailed publication. And I also will be using the textbook that I use in my class by Gerald Tortora, Microbiology and Introduction. So, unless otherwise noted, those are my sources, and I will throw in some of my own thoughts, of course, along the way. My background for this topic is coming from really when I started at the age of 15. I became interested in microbiology for the first time, oddly enough, by visiting Florence, Italy with my mother, who was a Florentine war bride. I went through a lot of museums and art galleries with her growing up, and there were paintings and images related to the Black Death, which occurred between 1348 and 1352 in Florence, wiping out half the population of Europe, about 60 million deaths. And Florence was the largest city in Europe at the time. That pandemic reduced the population of Florence from 120,000 people to 60,000 over a 4-year period. And that sparked my curiosity. And fast forward to the fact I ended up with a degree in biology And of course, this is the fundamentals of the biological principles of this virus. I had a great interest in micro after taking that class, fascinated with diseases and research in microbiology, particularly immunology. I studied under a professor at the University of Missouri at Kansas City who ended up at the University of Miami of Ohio, Dr. John Stevenson. I credit this presentation to him in honor of him, and also to Dr. Lewis McKinney, a professor of the history of science at the University of Kansas, under whom I did my history of science degree at the University of Kansas. So under Dr. Stevenson, I worked for the first time with viruses. We worked with T2 phage, a virus that infects E. coli, and I was fascinated with how viruses, you put one virus into E. coli and it produces 2,000 virions. So the burst rate is very interesting, and we don't know what that is for this SARS-CoV-2 yet, but that causes COVID-19 infection, but that is, a factor is related to its pneumonic effect. So I developed my interest in viruses at that time, took a course called electron microscopy. You're seeing here an image of the virus, of the corona cold virus under an electron microscope, same appearance as SARS-CoV-2, and got to see T2 phage under an electron microscope at that time, got to see viruses under an electron microscope. So foundationally, for those of you not in biology, you should understand that viruses are non-living entities. Bacteria are living entities. And viruses are just basically little replicating machines of nucleic acids. And they're very, very small, measured in trillionths of a meter, which is referred to as a nanometer. And so you can only see them with a special type of microscope. This microscope was built as a spin-off of World War II bomb research, nuclear bomb research. You're basically bombarding the image with electrons. It's a very expensive instrument. There was one in the physics department. There is one in the physics department at UMKC that I used. And so a lot of time for preparation. This is about a 350,000 times magnification, whereas you can see bacteria at 1,000 times magnification. So that gives you a little idea. And you can see this corona type of effect here. Those are the spikes sticking off the surface that gives it that halo, Latin term for halo, kind of like the solar corona type of effect around the virus particles. You can see 3 of them there in this yellow background. And then the scanning electron microscope on the right, about 130,000 times magnification, was developed by Cambridge Instruments in the 60s. It gives you it gives you the idea of the virus bursting from a ciliated columnar cell. These viruses infect the ciliated columnar cells. The virus is first discovered in poultry in 1937. There's just all kinds of species and animals. There's some very virulent strains, meaning more likely to be disease-causing in pigs, especially. That's why the University of Iowa and a lot of the state colleges are steeped in research in coronaviruses. Coronaviruses cause about 10 to 30% of all the common colds. Most colds are caused by the family rhinovirus, and there's 99 different antigenic types of that virus, so you can get a cold 99 times in your lifetime. By the time you're my age, you probably aren't getting as many colds because you're immune to the ones you've had typically in terms of the rhinoviruses. But coronaviruses seem to be a little different. There's quite a bit of evidence that you don't maintain your immunity very well to them, and sometimes they'll return. My guess is the more severe that you have it, the less likely that you're not going to be immune. You will be immune the more severe it is, if you survive it, of course. But the cold viruses normally aren't fatal, of course. They're in the nasopharynx. We're talking about viruses that come in through droplet transmission into the eye, or indirect contact into the eye, or nose or mouth. From 1 meter droplet transmission, which is considered direct contact transmission. There are 4 strains of cold viruses. So I want to start out by talking about cold viruses first, and then contrasting them with the 3 types of known pneumonic or severe forms, including, of course, especially I want to get to SARS-CoV-2, which causes the disease known as COVID-19. The NL63 is an interesting strain of the coronavirus. It is the most virulent. Sometimes you can get, some bronchitis from it, more severe types of colds than with the rhinoviruses. So the morphology is that the virus is typically listed at 125 nanometers, although there is a range, in this family. It is a spherical enveloped virus. The envelope is the orange in this image structure, in this cross section, and then the whole spherical artist's rendition of the virus. This envelope is what is particularly sensitive to your alcohol sanitizers. They inactivate this envelope, and so they're very effective. I'm not a big fan of alcohol disinfectants, for E. coli or staph, but they do seem to work pretty well on viruses. Remember, viruses are completely different entities than bacteria. And some of this is semantics, but the thing that needs to be understood from the start is that viruses are made of DNA or RNA, never both. Bacteria have DNA and RNA. Living things have DNA and RNA. So technically these viruses are non-living. There's no process that's typical of living things to them. They're just evolved in nature as basically diversity generating machines. So this is a single-stranded, in terms of the genetics, a single-stranded RNA virus. It's listed there. And some RNA viruses are double-stranded, which is unusual. This is a plus strand, which means it is, in essence, messenger RNA. When the virus is uncoded as it comes into the cell, this envelope is removed, and that pink portion is released into the cytoplasm of the ciliated columnar cell. It's uncoded, and it comes in, and it is read right on the ribosome, and the genes are translated into its proteins. So a negative single-stranded RNA virus would be something that is going to be requiring the virus to have a gene for another enzyme called an RNA-dependent RNA polymerase that converts the plus single-stranded RNA into a negative strand, and from there the virus makes its 5 main proteins that I'm going to mention here in this slide. So this is an RNA virus. About half of all viruses are RNA that infect humans. The other half are DNA viruses. The flu virus is an RNA virus. HIV is an RNA virus. And it is a 32 kilobyte genome, the largest of all RNA viruses. Consider the fact that probably has around 32 genes or so by my estimation, and HIV around 9 based upon this. I've not actually seen a listing of the number of genes, but they have been studied extensively. the coronaviruses in general. And what is unique about them is that they tend to recombine between animals and then go into humans and recombine, and then recombine and can go back into animals, and they change in that way. So not like your typical mutations where there's slight changes, they recombine, and the influenza viruses do that as well. So that makes them dangerous for big outbreaks. Although the flu virus changes every year, this one seems to be much more unpredictable about its recombination, and we'll talk about the biology of that some more in a later slide. The S gene, it codes for the spike, which is where you get the corona type of appearance on the surface. It is a sugar protein, a glycoprotein that attaches to the surface of the furin receptor on the ciliated columnar cells in the respiratory tract, and also the goblet cells, which are in the respiratory tract that produce mucus. HE gene helps with attachment, and that's the hemagglutinin esterase gene. It helps with attachment, and once inside for intercellular transport of proteins from the endoplasmic reticulum to the Golgi apparatus. I'll have a slide later that shows the biosynthetic pathway of the virus. The E gene is for assembly and release, and this is within the envelope. It's part of that. And another protein is the N protein, which codes for the protein capsid that surrounds the RNA of the virus, the plus single-stranded RNA, which is messenger RNA in essence. And then there's an M protein not shown in this picture that is in the envelope that gives the virus its shape and has some other functions that we'll talk about later. So with common cold coronaviruses, the mode of contact, the mode of transmission, is called direct contact. And technically, that's 1 meter. If you sneeze into someone's face, that's considered direct contact. And it's in the same category as having sex or kissing or anything like that. It's considered direct contact transmission, droplet within 1 meter. So it wouldn't be even to the camera. Now they're saying stay 6 feet apart. This is because of concern about potential aerosoling of the virus in people who really sneeze hard and don't cover. This is not typical. Anything over 1 meter is really considered airborne. This virus doesn't stay in the air typically, but in some rare situations it can aerosol and go further. So they're just as a precaution saying 2 meters, but that is not standard. Typically, this is considered direct contact, 1 meter. Beyond that is considered airborne, a different classification technically in mode of transmission. Indirect contact is what I would suggest, this is me, as the most likely transmission. People are sneezing into their shoulders, their arms, they're staying covered up. You're not sneezing directly on anybody at this point too much. Remember, culturally, you teach your kids, don't put that in your mouth, don't eat that off the floor, don't share drinking vessels, don't share forks, knives, and spoons, those kind of things. Wash the dishes, get everything off. Those are cultural norms. And so, in Asia, a cultural norm is if you have a respiratory infection, you get a mask when you go out. You don't sneeze on other people. They've learned to control this situation. This is something which is going to become normal, I think, in the United States. And so I think that people are going to be paying more attention around them when people are sneezing than ever before, or coughing. And so what I'm suggesting here is that there's a lot more transmission going on with touching the eyes. It's on your fingertips, you touch something, the next person sits down at the desk, touches the desk with their fingers, rubs their eyes or nose, and inoculates themselves. it's indirect contact. So when I go to put gas in the car, I get out of the car, I swipe my card, put it back in my billfold, put it in my pocket, punch everything in to do the gas, and then get the gas filler and put it in and fill up the car, hang it up, and then I disinfect my hands. I have to take the disinfectant out of the car with me and set it on the car. And someone needs to market some easy method on the wrist or pocket dispensers for these alcohol disinfectants going forward, so that we can go ahead and be outside, but and do things, but with the understanding that after you open a door, you're going to have to disinfect. After you touch things in the environment, you're going to have to disinfect. And you can't just stop and wash your hands with soap and water everywhere you go. There's no soap and water by the gas pump, for example. Cell phones are a terrible source of contamination, a common source of contamination, I think, with a virus that's this infectious. So it's unusual, the degree of infectivity with SARS-CoV-2, with COVID-19 disease. This is not typical of most cold viruses, aren't quite that contagious. But you get the idea. There's certainly a model for this. The virus survives in 2 to 3 hours on inanimate objects, up to 9 days in really intense fluids. We're talking about deceased bodies or something like that. It can stay in body fluids for a long time. So I think indirect contact is really big with this virus. Handshakes, hugs, and kisses, it's on the clothes. You touch it with your fingers and then you inoculate. They say the face, don't touch your face. Well, it's eyes and nose that we're really talking about, or mouth, sticking your fingers in your mouth, obviously. Most people don't do that too often, but children do, right? The good news is that the virus should go away in the summer, right? Because it's warmer. It's not because the virus doesn't handle higher temperatures. It's because people are more crowded indoors in the winter within that 1 meter zone of transmission. Think about basketball games, high school basketball games. Oh my gosh, they're so crowded. You pack into a gym. So this is an example of what happens in the wintertime. And in the summer, you're out, and so the virus tends to move into the southern hemisphere where it stirs up down there, and then it comes back the next winter to us, which is probably what's going to happen with this. Hopefully vaccines and treatments will be developed by then, so we'll, second time around, be able to do better. We hope it slows down in the summer. Of course, spreading this curve out is maybe going to run it into the summer to some extent too. Coronavirus is not normally transmitted by blood, feces, or urine. This virus doesn't really get into the body fluids. It stays in the respiratory tract, which is part of the reason you don't get a real strong immunity to it. The immunity you have is non-specific alpha interferon. You don't get a lot of T and B cell clones being selected, interleukin 4/6, plasma cells, memory cells. All of that immunological specificity is not kicked up by coronaviruses because they're in the mucosal region where immunoglobulin A is produced by the goblet cells. There's some immunity, but unless you get a really bad infection, is the sad flip of this, you're not going to get good immunity. If you survive a bad infection with it, you're probably going to be immune to it the rest of your life. If you get it and you knock it out with alpha interferon, you're not going to have that long-term immunity. So you could get it when you're older still, again, because immunity rolls off as you get older. So, it's not transmitted like in the case of my further research for my master's degree in the history of science. I spent about 4 or 5 years studying the origin of HIV, studying the first decade of AIDS in Europe. I wrote to World Health Organization and Center for Disease Control specialists behind that Iron Curtain at that time. Scientists cooperate with one another. I sent surveys out and I got lots of good feedback. My work was very important at that particular time because I was getting information from Iron Bloc countries as to how the AIDS pandemic was spreading there in the late 80s. This was at the University of Kansas with Dr. McKinney. I was very fortunate to have that opportunity. I got responses from even Anthony Fauci at the time for the American side of it, and also from Jonathan Mann, who is the president of the World Health Organization, responded to me. So I had a long interest in pandemics. So that was the one that I did for a master's degree in the history of science. I later published an article on the 6 theories on the origin of HIV. Evidently, the virus jumped the species barrier from monkeys in Africa. There's been some conjecture about that, but that seems to be where the evidence has landed. Later, I went on for my PhD, and I took a course at the University of Missouri, Kansas City called Black Death. And I was just thrilled to be in that class. I was all PhDs in history. I was the only science major in there. A fascinating class. And we read texts that had been converted from Latin into English about case descriptions and so forth at the time. And of course, a lot of confusion in the 14th century as to what was going on. But I ended up publishing an article that based on a paper I wrote for that class about how the disease was transmitted and the etiology of that disease being Yersinia pestis. There was a mutation in a bacteria at the time that caused the spread, and the fact that it could be spread so many different ways. Well, the coronavirus is only spread by the droplet, and that indirect contact route, whereas the reason that caused such a horrible pandemic and killed half the planet was because it was contaminated by food, water, airborne, flies, zoonoses, insect bites. It was just all over the continent, getting on ships. You couldn't get away from it. Everybody was getting infected, all ages, no matter what you did. They had no clue. You go out to the countryside, you get infected. You go into the city behind the wall, you get infected. You don't have any contact with people, you still get infected. It was, to some extent you could do isolation, but it wasn't understood what was going on at all. Of course, there were no treatments, and it was very hard on the medical profession at that time, creating a reduction in respect for medicine at that time in Europe, and changing the way people looked at life considerably. So this appears to be jumping from animals into humans. So it can do that, but that's very rare. And that's what starts the pandemic, is the fact it can recombine. But it typically doesn't do that. This is crowded conditions with animals, lots of different kinds of animals in markets that are live. People like live animals. They're fresher, right, to butcher. And also, the combining of fluids of these animals in markets, crowded markets, lots of people handling them without gloves and those kinds of things. So that's how these things can kind of occur. And the World Health Organization has put the kibosh on it, but, as much as possible. But it's hard to regulate because there's a black market for these animals. The pathophysiology of the cold virus is that it infects the ciliated columnar cells, as I said, and makes many thousands of copies. We don't know how many it makes of itself. The symptoms of the virus are, as you know, inflammation and mucus buildup in the nasopharynx, sneezing, coughing, normally not severe. But in people who are of low immunity, immunocompromised or immunosuppressed, which I'll talk about more when we get to the pneumonic forms, the typical situation is that you get a primary viral infection, and then you get a secondary bacterial infection, such as with Streptococcus pneumoniae. At first, the solutions are clear with a cold virus. You get clear mucus. That's when you're still in the cold virus phase. And that will go away in a few days due to the production of alpha interferon. But if it stays more than 3 or 4 days, you're into secondary complications. It's thicker, the mucus is thicker, it's turning colors, brown, yellow, red, green, different colors and thicker. Then you're into secondary infections, red in the case of strep pneumo. So Streptococcus pneumoniae is the most common cause of death in the United States from a bacteria, and it's in the elderly and retirement homes, right? That's the way a lot of people die. Start out with a cold or the flu, it depresses the immune response, they get this secondary opportunistic infection with the strep pneumo that's in the lungs, it forms a capsule from all the extra nutrients that are available. This is standard fare in retirement homes. This is the way people are supposed to die naturally, so to speak. The old man's friend, it's sometimes called, strep pneumo. So it starts with a cold virus. And so coronaviruses have been, shall we say, terminating life for the elderly for a long time. What's different about COVID-19, of course, and these pneumonic versions is that you don't need the strep pneumo. It goes straight for the lungs. This is called atypical pneumonia, and usually it's not as severe. This one happens to be, but usually it's not as severe as with a bacterial pneumonia. Incubation period's really quick, 2 to 4 days. So that's the cold viruses. You get good immunity, usually, and it goes away. Alpha interferon's an interesting substance. It's part of non-specific immunity. Everybody produces it in response to viruses. It is a function of, it's a substance produced by any cell that's infected with a virus. Tells the neighboring cell to build antiviral proteins that basically surround and block that cell and that virus from getting into other cells, and that puts an end to the infection very quickly. So the more acute the infection, the quicker it's shut down, classically, as a part of the way the immune system should work or does work typically, and you're usually immune to it the rest of your life. That's called self-limited. That's the way it's supposed to work in healthy people, right? That's the way the immune system works. You don't need a vaccine for cold viruses because all you need to do is get a little rest, let your alpha interferon do the job. Maybe you reduce some of the symptoms with acetaminophen or naproxen, drink plenty of water, have a humidifier, take a hot bath, definitely avoid smoke, definitely avoid dairy products which increase mucus, and whatever you do, don't exercise because you need the protein to build antibodies to fight the infection, and not build muscle at that particular time. A lot of people like to exercise when they're sick because they think that it makes them feel better. These are people who exercise routinely, of course. But yeah, you shouldn't really exercise when you're not feeling well. You shouldn't be out in public exercising, definitely, and spreading the virus, right? So now we get to the pneumonic form. Contrast this with the cold viruses. These are the SARS, MERS, and SARS-CoV-2 that causes the COVID-19. So these are recombinant intensive viruses. They tend to have recombined in animals and in these markets. They go from animals to humans and back to the animals, and create new strains that stick sometimes, like it has 3 times in the last 20, 30 years. And this is all a function of crowding animals together with humans. I mean, there's no question about the fact that that's how these things occur. These viruses are more contagious. The mutations that actually occur that are a part of the recombination are going to lead to a more efficient adherence and fusion into the cell, into the ciliated columnar cells. They're going to probably have a higher burst rate. They're probably going to have a lower virion infectivity threshold. And also what's interesting about COVID-19 in particular, and I'm not sure that SARS and MERS do this, but I think I did read it with SARS, is that the virus doesn't just burst out into the respiratory tract and then spread down into the bronchi and into the lungs. It spreads cell to cell. Okay, it's fusing cells together that way, so the immune system doesn't really see it In that way, the specific immunity doesn't recognize it. Your macrophage aren't picking it up, this is a fairly unique feature, I think, of these pneumonic versions, and certainly CoV-2 seems to be doing this, and SARS did this. Now, the worst news about this one, SARS and MERS, according to all the evidence, were not, that should be MERS, not Mars. SARS and MERS were not infective until symptoms began. The incubation period is the time period from when you're infected until you show symptoms. Those viruses weren't transmissible until symptoms began. This virus seems to be transmitting SARS-CoV-2 that causes COVID-19, the current pandemic, is transmissible slightly before. We don't know how many days before. Certainly when you're sneezing and coughing and mucus droplets are available occasionally before full-blown symptoms start, you are transmissible then. One of the false things that's out there is that people don't have any immunity. It's a novel virus, but you do. You get pretty good immunity from this. Still, it's in the respiratory tract. You're not getting that classical immunity because it's not in the lymphatics. It's not in the not getting into the bloodstream as much. It's not a bloodborne pathogen like Ebola or something. You're not getting great immunity from this that lasts, but you do get great production of alpha interferon if you're healthy, and that puts the kibosh on it. So 4 out of 5 are self-limited. 4 out of 5 basically experience this like a cold or less, just a typical cold. You get rid of it real quick, it's gone in a couple days. Nasopharyngeal involvement, no thoracic involvement, no bronchitis, no lung involvement whatsoever. 4 out of 5. And that's with COVID-19. This is well documented. And about 20% have serious symptoms, okay? If you do the math there. Now, what are serious symptoms? We'll get into that. Obviously, tightness in the chest, difficulty in breathing. So out of those, 5% are hospitalized with respirators, and according to the CFR, the case fatality rate's 3.4% coming out of the World Health Organization. I haven't seen an update on that being changed. Immunity consists of alpha interferon, immunoglobulin M, and phagocytosis, immunoglobulin A. Huge debate about chances for reoccurrence and immunopathologic effects. The flu virus depresses the immune system for about 24 hours. It affects the mucociliary escalator, the movement of mucus up towards the epiglottis from the bronchi. It freezes that, much like cigarette smoking freezes the mucociliary escalator. We don't know. I have not seen anything good on the pathophysiology that there's been enough research to know what the immunopathologic effects are. In other words, does it depress the immune system? They've shown in mice that SARS reduced the number of T cells. So there's something going on that is serious with these viruses as to why it's spreading into the lungs so quickly. So there's a hit on specific immunity going on here, which would explain why you could possibly get infected with it again next year, next winter. Survive it this year and get it again next winter. Again, my general view of the way the immune system works is if you get sick from it, you're immune to it. That's the way it's supposed to work. But obviously, we're getting exceptions to that rule already being published. So there's also a big debate about whether or not there are carriers for COVID-19. That won't come out until you test everybody. I mean, if we tested 330 million Americans, we would know, right? And by next winter, we're going to have availability of tests, hopefully in our home, where we could test ourselves and report. And that way we'll know if there's carriers with this. The good news is SARS-CoV-2 that causes this COVID-19 is the lowest mortality rate of these three pneumonic coronaviruses. That's fortunate that, it's just affecting people that are immunocompromised, immunosuppressed. But of course, since it's very highly more infective than both of those, that's the bad part of it, is that it's a higher rate of transmission, and those factors are the big concern here. So who do we look out for? Well, that's a big slice of our population, especially in the United States because of the obesity epidemic. Those people are at risk. They haven't mentioned that issue. I'm going to throw it in. I teach nutrition. I think people who don't eat right, don't exercise, are at risk here. I think they're at greater risk. Not as much as a smoker, not as much as if you're elderly, not as much as, if your immune system is rolled off, the immune system starts to roll off about age 40, your clones of T and B cells. So when you're 70s and 80s, you're at a greater risk for reoccurrence. Example, shingles comes back. You're exposed to VZV, varicella zoster virus, when you're 3 years old. You get chicken pox. Usually you get rid of it completely, but some people it's dormant in the thoracic ganglia, and 30%. It comes out when your immunity wanes. What is that, about 60 some years? So yeah, the longer you're away from that initial infection, the greater the chance for reoccurrence. But certainly age is the most natural way to be immunocompromised. Those people are at the greatest risk. They're saying over 60. Depends on how you take care of yourself. 60 is the new 50, right? 70 is the new 60. So, I'm looking at people in their 70s, definitely 80s, 75, 80, you're starting into recognizable immunocompromised status, regardless of how healthy you've been. So that's just the natural aging process. Lack of exercise, poor diet, that's my thing. I'm throwing that into the equation. I do not understand why it's not emphasized more. This is an opportunity to bring that issue out. Or certainly if you have an immunological disorder, an autoimmune disease, you're at risk here. Especially if you have any kind of respiratory disorders, asthma, emphysema, heart disease, stroke, all of that has been listed. Diabetes certainly is a factor here. But what's interesting about the South Korean data is that this is young people who smoke cigarettes. Males smoke cigarettes in South Korea, and so many of them have died from this. And here you think about people who use the nicotine forms, all these new instruments and tools for getting more nicotine into the system. I think those young people are going to be at risk. We have an epidemic among young people using enhanced methods for intake of nicotine, not just cigarettes. So that's a whole different bag. Immunocompromised. Immunosuppressed would be, I think a big factor in risk is cigarettes. In this case, you're immunosuppressed, right? You're suppressing your immune system by something you're taking, illegal or legal types of medications. Taking meds or treatments to suppress the immune system, like chemotherapy for cancer or radiation. These people are at great risk, right? They don't have a normal immune system. And so if you add up all the people that are immunocompromised, immunodepressed, if you will, or, which is a natural sort of thing, right? Or immunosuppressed, which is due to medications for diseases, that's a pretty big swath of the population, and I'm sorry to say that I agree that could be millions of deaths in the United States. I mean, we're ripe for this. We have been ripe for a long time. This is not something that my former students have been teaching 26 years haven't heard from me in every lecture for 26 years. Been preaching this for a long time, and so have many other microbiologists. I find it very frustrating that somebody, the richest man in the United States, who has his own foundation to prevent diseases and pandemics internationally, Bill Gates, donates money, and Bill and Melinda Gates, the foundation, to fight malaria and other diseases that are pandemic in nature, can't seem to get through to the government. I mean, what can I do as a community college instructor more than teach my students about it? I published articles on this issue. I predicted in 2014 when I did a presentation that this could happen. I mean, I've been preaching it all around, writing authorities, talking to people through the years. If Bill Gates couldn't get through, you basically need the military to come in, and when something like this starts, you just shut down that area. Nobody can get out. You find out what it is. You quarantine that area. That's what they're doing in Asia. These are the lessons that you learn from China, and they stopped MERS, they stopped SARS before it got to be all over the planet in a pandemic form. They're ahead of us in their understanding and their cultural adaptation to these diseases. This is a huge lesson for the United States. We're a country that loves freedom. You have a choice whether you can take medications. You have a choice as to whether or not you want to stay at home or whether you want to cough all over everybody and go somewhere. It's your choice. That's not the case in other countries. They go in and they shut down the perimeter with the military, and that's a very scary proposition in a democratic free society historically, based on founding doctrines, right? So this is a big concern, but it's a pandemic. It's spread to different countries. Technically, by definition and numbers, it's not, but they're trying to prevent it because they know it could be. But going through the United States like this, we're extremely vulnerable. We have 27 million people without health insurance, and I guess we're going to just insure those people, but that's just bizarre to think about. So SARS, 9.6% mortality rate, much higher mortality rate than for COVID-19. Not transmissible until symptoms begin. This thing goes right for the lungs, kind of bypasses the nasopharyngeal and goes on down. Again, mostly killing the elderly, same kind of treatments, transmitted by the horseshoe bat and Asian palm civet cats. That's where the recombination was found. This has been well documented, this beautiful animal in the wet markets. Wet markets meaning live animal markets. And the horseshoe bat seems to be the main source. It's used for meat, a meat source. Most all the viruses are coming from horseshoe bats. You see that as a consistent factor in this recombination of the virus. It's not really so much mutation as it is recombination in this particular virus that's the issue. So MERS, Middle East Respiratory Syndrome, originated in Saudi Arabia with dromedary camels, again, and horseshoe bats mixed together, undercooked camel meat, unpasteurized camel milk, and camel urine, which gets us to the fact that many of these collectible items from animals in various cultures are a function of cultural myths, cultural traditions that have been around a long time, anti-aging ideologies. Witness rhinoceros horns, elephant tusks, camel urine, and pangolin scales. A range of things that are associated with the World Health Organization trying to stop basically leading to the extinction of animals in some cases. So these are traditions that are, trying to be changed by the World Health Organization, that are cultural in nature. We have our own cultural issue, right? Right? We don't do anything about the spread of viruses, and we're having to learn about how to do that. We don't disinfect often enough. We have contact with people a lot after infections begin, and we're learning that you have to take a different approach. The incubation period is 6 days. Up to 14 days is rare, but note it's not transmissible until symptoms begin. Terminal cases with kidney failure, pericarditis, and disseminated intravascular coagulation makes it a little different. And there's diarrhea associated with MERS, more common, and we're seeing gastroenteritis, diarrhea with COVID-19 as well. So you get to COVID-19 and the symptoms are shortness of breath. You've heard that. It's a clear sputum typically. We're seeing some yellowing in some cases. You see reports of that. Lung x-rays give a broken glass appearance. Tightness in the chest. Gastroenteritis is sometimes triggered indirectly. The virus is not going into the GI tract, but the depression of the immune response can lead to this disruption of the normal microbiota in the GI tract. So it's telling me that there's something immunopathologic probably going on with this virus and the reason for its high mortality rate. Survivors sometimes have delayed sequelae. They haven't really reported too much on this because we're not on that side of the infection yet, or the pandemic, but some of the people who have survived are having terrible side effects after the fact. Type blood A seems to be most susceptible. I don't know what the correlations will end up being with that. Incubation period 2 to 14 days, 5 to 7 day average. Transmissible before symptoms, again 2.2 infectivity rate compared to 1.3 for the flu. So there's something about the burst rate of this virus or the fact that it gets into cells more efficiently that's at work. The origin is probably horseshoe bat and pangolin. This is images of the pangolin, an amazing animal that is the second most trafficked animal in the world after humans, more trafficked than elephants and rhinoceros. The scales have medicinal purposes and the meat's edible. 2.7 million pangolins killed each year. It's on the endangered species list. Looks like the origin is a mutant novel strain, combination horseshoe bat and pangolin. The first case now reported near Wuhan, 11/17/19, unrelated to the wet market strain. Two slightly different, not really strains, subtypes of the virus, and in essence then two simultaneously emerging versions, but that shouldn't be a problem for a vaccine. They're not that different. The epidemiology, is changing day to day dramatically, but we're the worst affected country. Over 1,000 deaths, 3.4% case fatality rate, as I mentioned, and the Italian situation. And the fact that it hasn't really gone into any of the big countries yet in terms of population. India being the second most populated, Indonesia third, Pakistan third, fourth, however those flip. South America is going to get it now with the winter coming. So it hasn't really gone around the world yet. This could be and come back next winter and not really ever stop, just keep going unless we come up with a vaccine or medications for this thing. The infection fatality rate is an epidemiological calculation that requires estimating undetected infections. And the IFR is estimated to be 1%. So it's not that 3.4%. In South Korea, where the most rigorous testing occurred due to a test provided by the World Health Organization, it's 0.65%. It's interesting to compare COVID-19 with the flu. We're in a 100-year anniversary of the pandemic of 1918 to 1922. It's called the Spanish flu, and that is not correct. They just happened to recognize it in the literature at the time. But it probably originated on a pig farm in Kansas and in a soldier who trained in crowded conditions at Fort Riley, and the soldiers there took it to Europe and spread it there. So It's almost that one is incorrect to call it Spanish flu. It did not originate in Spain. We don't know for sure where it originated, but there's some evidence it started in Kansas, interestingly enough, certainly a part of Kansas microbiology history. So interesting that we're on a 100-year anniversary for the influenza pandemic. Now, here's the interesting thing that's not being discussed to me. This was an H1N1 strain, we now know, that killed 22 million people over a period of 4 years in the setting and return of soldiers back to the United States from the trenches in Europe at the end of World War I. And it spread all over. It killed 26,000 people in the greater Kansas City area, killed a lot of young kids, elementary, junior high, high school kids in Strawberry Hill at the time. There's the museum, Strawberry Hill Museum in Kansas City, Kansas, has a room dedicated to the influenza pandemic. It was a horrendous event. But here's the thing that's not being mentioned. Actually, what killed people was a bacteria that caused a secondary infection. Started out with flu, depressed the immune system. This was an antigenic shift virus that very little herd immunity, previous recognition, minuscule, and so covered the whole planet, went around the whole planet 4 times before it, got stopped, and it for 4 years. Literally goes around and infects the entire planet 4 times. But what was going on that piggybacked on top of it is what's not being mentioned. There was a bacteria that mutated at the same time, coincident with that, called Haemophilus influenzae. And this is what they thought was the cause of the disease. They never did discover the virus at the time. It was a few years later before they discovered that it was a virus that caused the flu. They knew it started as flu symptoms and ended up in meningitis. Well, they thought that was the cause of the flu. So this bacteria is intentionally misnamed in honor, historically, of the futility and the struggles of medical professionals, scientists, researchers who gave their lives, and the people who died from this virus, never knowing what caused it. And the lesson that I think is important, really the bacteria should be called Haemophilus meningitidis. They won't change the name in honor of the deceased. So this is the only bacteria that's intentionally misnamed. It doesn't cause influenza. It causes meningitis. But it happened during the influenza epidemic, which, the symptoms started out like the flu. So I think there's 2 dangers here. One, that COVID-19 that virus could recombine again during the middle of this pandemic before it's shut down and become more virulent and kill people of all ages. Or there could be a piggybacked version of the bacteria, of a bacteria or a fungus or anything that mutates and becomes virulent on top of it. Remember, the Black Death was a function of a mutant bacteria. The last pandemic 100 years ago, we're celebrating by having this pandemic now, recognizing what these microbes do. What microbes do, I've heard it misstated by many people who don't specialize in microbiology, that these microbes don't mean to make you sick or hurt you. That's a mischaracterization. They don't think about anything. They're just creating genetic diversity. Viruses in particular do that. And what does appear to be occurring as a part of biological evolution is that they cull the population. They keep the various living forms on the planet balanced between each other as in their own ecological niche. Okay, that's a very important concept to understand. These viruses are trying to cull the population to half of what it is for Homo sapiens because it's encroaching on all of the other living things and causing death to life on the planet, as it were. That's just the nature of the way they work. About 10% of all bacteria are pathogenic. I don't know with viruses. There are so many viruses. There's gazillions in the ocean, right? Viruses are specific. They infect everything specifically. So, you can't get a virus from something else that infects something else typically unless it jumps the species barriers. You're actually seeing evolution, biological evolution at its point when you look at these three mnemonics. Huge biology lesson. That's the lesson of the day. Haemophilus influenzae causes meningitis. It was a mutation that piggybacked on top of an antigenic shift influenza virus. That's how complicated this is. That's how multifactorial the biology here is. That's the lesson. And we're not through this yet. If it's just COVID-19 and the virus is stable, we'll be fortunate. We'll be very fortunate because by next year we should have some things going against it, hopefully. So, look at the comparisons. Even the flu in 2018-19 killed 61,000. In 2017-18 it killed 80,000. We're at 1,000 and we're freaking out, and we should be. I'm just saying that the flu virus does this every year. It mutates annually. And the vaccine wasn't that effective this year. The good news this year was that that the virus didn't kill very many people. It's been a mild flu season. If it had been a bad flu season, then the coronavirus would be, in essence, piggybacking on the flu. That could happen next winter also. So there's a long ways to go, and there's a lot of fear here. I didn't mean to generate all these fearful aspects, but I want you to understand the reality of this. So you're better armed to understand what we're up against. Best to know your enemy, if you will. And the last pandemic actually was 10 years ago, roughly, with the H1N1 influenza virus that originated in Mexico instead of China. Why do most things come out of China? Because they're the largest population of humans in very crowded conditions. Markets, open markets, that part of it can be changed. That part of it needs to be worked on. World Health Organization is trying to clean it up. Very difficult to clean up the Ganges River. It's a religious tradition, right? With parasites. That's a huge issue for the World Health Organization. How do you get people to change their cultural norms? How are we getting people in the United States to change their cultural norms? Now we're the ones responsible for spreading it. You see, the microbes find a way. They find a weakness in each cultural system. It's very interesting culturally for how it goes through different populations. So what does this virus do? It replicates and kills ciliated columnar cells. One are the ciliated columnar cells. This is the cilia on the columnar cells that move dirt and dust up against the forces of gravity. They wave up, pulling dust and dirt out of the bronchi up towards the epiglottis, where you then swallow it. So as you're swallowing, it takes about 24 hours to go from here to here, to move up to the epiglottis. So if you smoke cigarettes, you freeze the mucociliary escalator. Does that tell you why people die from COVID-19? It's as simple as that. You're not getting the removal of mucus that normally would occur if you're a cigarette smoker, right? And it's addictive. And the nicotine portion is addictive, but it's the smoke particulate that's causing the freezing process. And so, by the way, the flu virus freezes the mucociliary escalator, and that's a part of its non-specific immune hit that it makes. So the number 2 shows you how big these columnar cells are. They're pretty big. This is a histological cross-section. And number 3 is the goblet cell. So the virus is not just in the respiratory tract. It is going between cells during a fusion. Cell-to-cell fusion is also occurring. Immunoglobulin A is the antibody that's secreted by the goblet cell. That's the immune side of it. But this is the picture of the respiratory tract and the actual cells with a staining process that allows you to see the cells. So the virus S, the spike protein, is going to fuse with the goblet cell and the ciliated columnar cell at furin. Furin is a protease that allows the virus entry. S2 then splits the fusion peptide and gains entry into the cell. So first the spike is cleaved, and then S2 is the stem of the spike, and that allows it to enter the cell, sort of melt into the cell. The enzyme furin is abundant on respiratory tract cells. With SARS-CoV-2, both S1 and S2 are uniquely cleaved on viral exit, so they're ready to go, which is different. That's where the mutation occurred with this virus, is that think of it as when the virus leaves after it's infected a cell, when it leaves, it's already got the keys to get into another ciliated columnar cell. Most of the time, coronaviruses, once they get there, have to call a locksmith and let them in, which is a little bit of a delay. This is quicker, and it's twice the infectivity, apparently, because of that. So the SARS-CoV-2 is theoretical doubling of infectivity. Note, this entry mechanism is not found in bat or pangolin strains, so zoonotic origin has not technically been discovered. One well-known causing cold virus, L63, which is what SARS apparently jumped from, gains entry with the ACE2 angiotensin converting enzyme cell receptor, and so does SARS-CoV-2. So both of these all three of these viruses gain entry via this mechanism. So there's two ways to get in, not just the spike binding to the furin, but also the spike binding to the ACE2 receptor. So two different mechanisms of entry. So in terms of it being a replicating machine, this is from a 1997 image, but of the cold viruses and the way they work. And basically it's the same way, except that, we have a virus that in this case is getting in a little differently. But once it's in, it's interesting that shown here, the red positive strand, the plus single-stranded RNA, is red on the ribosome, and that produces the RNA-dependent RNA polymerase, which then is going to be able to convert the positive strand into a negative strand, and you get the production of the 5 major proteins, and then the positive strand is made back again for the purpose of packing itself into the capsid, and then the M protein is inserted, one of the envelope proteins is inserted into the endoplasmic reticulum that facilitates, the M protein facilitates entry into the endoplasmic reticulum. And then you have assembly and package of the virus in the bottom right-hand corner in the endoplasmic reticulum. And then the spike is synthesized, and on its way out, the virus takes the endoplasmic reticulum with it again. So it's shrink wrapped in this phospholipid bilayer of the endoplasmic reticulum on its way out. So it's got a double layer of phospholipid, which allows it to fuse into the cell membrane and then pop out into the extracellular space, and it sheds that phospholipid bilayer that it picked up from the second time it went through the endoplasmic reticulum membrane. That's basically the idea of this biosynthetic pathway. Obviously, a great deal of detail is understood about the nucleic acids. You can find it in the article from the Iowa State or the University of Iowa researchers that I mentioned on the first page. to get more information about this, but an extraordinary detail and well-known mechanisms. And the idea here is to understand this so that you can make medications to block these various steps, and we're going to be talking about a few that do this. So the diagnosis is the polymerase chain reaction based on snippets of SARS-CoV-2, pieces of the messenger RNA that match up with sequences in the Thermo Fisher PCR that's prominent in hospitals in the United States. I recommend Bianca Negredi's The Scientist 320 article. She does a nice job of summarizing where the cutting edge is. By next year, I'm thinking we should have ability to paper strip, McGovern Institute, MIT production of being able to diagnose and test in home by next year. So what's the prophylaxis? 40 giorni. In 1377, the Venetians wouldn't let a ship in for 40 days because of smallpox spread. They waited, they took food out to them, but they wouldn't let them in because they wanted to make sure they weren't bringing it from one of the cities further down the Adriatic. And, we have a 14-day quarantine. So the term quarantine comes from 40 days, which is historic Italian thing. We are again holding ships off the coast and now holding flights, and that is really the best way to put the kibosh on it. Social distancing, the idea is to flatten the bell-shaped curve of rate and frequency of infection, pure epidemiology. This allows more time for health workers to respond, get equipment in order to fight the influx of the virus. And of course, Dr. Anthony Fauci has made himself famous again. He was already well known to me for this. Alcohol hand sanitizers, Corona beer doesn't work. Face masks are best, especially if with a respiratory infection. Wash your hands with soap and water after touching public surfaces. No contact, especially with at-risk immunodepressed or immunosuppressed populations. There's no evidence, but the Chinese mention 125 different substances found in food that are furin inhibitors. So everything from broccoli to oregano to oranges have these things in them. Now, how they would work if you eat them, how are they getting into the respiratory tract and having an inhibitory role, I have no idea. There's no evidence they work, but these are good foods to eat anyway. I advocate, of course, a whole foods, plant-based diet and regular exercise to prevent you from getting this disease. And the immune boosters would include sleep. Sleep is an immune booster. Garlic, green tea, astragalus, sunlight, shiitake mushrooms, curcumin, liposomal vitamin C, and parsley. Parsley being very high in vitamin C. Can't hurt, right? Seems to be some evidence, but not entirely worked out. But all of these treatments are just supportive, not curative. Even the ones that have been developed, the remdesivir is a nucleotide analog that I talk about in my class for adenosine that blocks the enzyme necessary for viral replication, the RNA-dependent RNA polymerase. Gilead Sciences. Probably a good company to invest in, right? And favipiravir is a similar purine analog that blocks RNA polymerase activity, and this is being tested in Japan already by Toyoma Chemical. And then, they've tried stuff on SARS and MERS. Ribavirin with alpha interferon together seems to work a little bit. Keep in mind, this is my prop, my viral culture dish. You put fetal calf serum in here, monkey kidney cells, you put your virus in there, and any coronavirus will kill monkey kidney cells, right? Especially SARS-CoV-2. But if substances which seem to work in here to keep the virus from killing the monkey kidney cells include, these substances. So in the lab, they've shown that chloroquine and some of these things work. They're antivirals because of that. But they haven't been tested in humans sufficiently. And obviously, this is where everything is going. You test it in mice, you test it in rabbits, guinea pigs. You test it in animals to see what it will do, and then you try these out in humans. And clinical trials are already beginning with some of these in humans. Wang, Cell Research, good article, recommended. Antivirals to interfere with ACE2 receptor, antivirals to interfere with the furin receptors. But the biggest issue is just pragmatics, right? There's not enough tests, there's not enough hospital beds, there's not enough nurses, there's not enough N95 respirators and ventilators to go around. We are swamped, and we didn't even have a bad flu season. What about the vaccine? Moderna vaccine, Seattle, you've heard about it, I'm sure. They've been talking about it a lot on the news. This company is, of course, a big push from Bill Gates. He's very optimistic. This is a platform where you have RNA viruses that you can RNA sequences that you can change to fit whatever virus pops up. You're ready to fight an infection, an outbreak right away. Vaccinating the population around that site is what you want to be able to do right away. And that's what's coming with this. Instead of your traditional live vaccines, although we may still end up having to need live vaccines, can't rule that out. Those are more dangerous and require many more years of testing. These are probably not going to trigger any disease recombination. What they do when you inject a piece of RNA into a human that has the same sequences as the proteins on the SARS-CoV-2 is that you get your body to make the little proteins that are the same as on the virus, so that creates an immune response to the virus. You get production of immunoglobulin A, and it's right out there and neutralizing the virus as it's going through the respiratory tract. This has some potential, and that's the future of it. It looks like. We'll see. We hope before a year, right? Because they're already testing in humans. The good news is this virus doesn't really mutate much. Still, the risk is that it's going to recombine. Slight mutations. There isn't much difference between the virus in Italy and China and in the United States. That is good news. And the fact that we can, with the Global Virion Project, check for these things. We couldn't do that with HIV so easily. Different COVID-19 infections can be RNA sequenced very quickly to determine the mutation rate. The virus has changed very little, as I mentioned. So this is good. And there's an adjuvant company, GlaxoSmithKline, is working on a vaccine that boosts the other vaccines. So probably both of these together are in the future. It uses the M protein, which is used in assembly and mediation of molecular mechanisms. So, one of these Moderna mRNA sequences, along with this adjuvant vaccine, looks to be in the future. Keep in mind, you don't have any immunity to COVID-19 just because, like me, you've had probably all 4 of the coronaviruses in your lifetime. So we can get into a lot of history lessons here, a lot of sad aspects to it. Climate change is interwoven into pandemics, involving Aedes aegypti mosquito, Zika virus, dengue fever virus. We didn't get those up here. So we just tend to ignore it because we don't get the big mosquito hits from malaria and those types of things that kill many more people. Cholera due to natural disasters. We've never really been through that. But these are the lessons. Overuse of antibiotics in cattle and pigs, 65,000 deaths a year. Most of the antibiotics are given to animals. The bacteria that are antibiotic resistant get into humans, and then they don't work when we need them. 27 million people uninsured. I have to say that healthcare is not a privilege. It's a human right. It's time to bring that out. This is bringing that out more clearly than ever in an election year. It's amazing to think about the fact we don't have any health insurance. Other countries understand this, that you have to have everybody covered because that stops pandemics. Malnutrition. I could go on for hours, right? the United States shouldn't have cut funding for the Global Health Unit, but they're not listening to Bill Gates. Why would they listen to me or anybody else, right? They cut funding to this. My former brother-in-law was on the Global Health Unit of the Pentagon after the Ebola epidemic. We should not have decided to wait for American companies to generate PCR tests. I saw this with the AIDS pandemic, the same kind of thing, where we didn't have cooperation on a level that's needed in industry and government to keep the virus from spreading at that time. But we should have accepted the World Health Organization tests instead of wanting to promote businesses in the United States. That's a function of the type of leadership, right? And so we're ripe for this pandemic because of the type of leadership, although the leadership has never really been where it should be on this, coming from Congress, executive branch. We're not getting a clarity of understanding. This doesn't have anything to do with politics. It's the nature of the beast that we're dealing with here. And it's all about everybody. It's not Russia, United States, China, Korea. It has nothing to do with that. This is an area where we all have to work together. It's amazing for that. That's the interesting sense of humanity that it has, that it is recognizes our diversity. At the same time, it unifies us in coming together over a different kind of war that we're all in. Pandemics are not just a health risk. They're disruptive to the global economy, stock markets, and the banking system. And we don't budget for them, like natural disasters. I mean, you spend $150 billion and you could have prevented this $2 trillion disaster. And it's not over. It could be more than that. And obviously, xenophobia has to stop. I mean, I did a presentation on behavioral disease anthropology. The United States has plenty of behaviors that are conducive to pandemics, that support pandemics. Every country's got its thing that creates that niche that promotes the sense of pandemic. But I tried to point out in my presentation in 2014 in my academic symposium how human behavior increases the rate of emerging infectious diseases. With the Black Death, 1348 to 1352, everybody was blaming each other for what was the cause. Same thing with syphilis. It originated in native populations here, and Columbus had brought it over. The Columbian Exchange, the influenza pandemic, we call it the Spanish flu, the AIDS pandemic. The Russians blamed us, we blamed them. It went on and on. It's just been always like that. And the blame game is as old as human behavior. And extremely counterproductive. The best book that I've ever read is Plagues and People. You can get it for $1.50 on Amazon as a paperback. The Whole History of the World Based on Infectious Diseases, 1976. Incredible historian William McNeill. And Nathan Wolf's The Viral Storm, 2017. The only book I read that predicted the coronavirus pandemic that we've got here now. I'd like to thank Dr. Zagovich, Yelena Zagovich. I would like to thank Kansas City, Kansas Community College for allowing me to teach nurses all these years, pre-nursing students, their last stop course before the program. I'd like to thank Tom Grady, people in the Center for Teaching Excellence, the Learning Commons, the librarians, so many people that have helped me out, my biology colleagues, my deans through the years at Kansas City, Kansas Community College. I'm wrapping up my career at this time, and it's been just an incredible privilege to teach at Kansas City, Kansas Community College, a great community college. I don't know how many thousands of students I've had. I've taught 2 micro lectures and 2 micro labs for 26 years, and in the summer as well. And I can't go into a hospital without seeing somebody I know that was a former nursing student. And it makes me feel so proud to know that these people are on the front line I admire them tremendously for their dedication. I used to joke about nurses a lot, but truly, in fact, we're finding them to be, the heroes in our country, and a lot of the retired nurses that are volunteering. It's very heartening in that respect that people are coming together as a result of this in a novel sort of way. Again, thank you very much for this opportunity to make this presentation. If you would like to contact me with questions or a conversation or critique or analysis, always appreciate feedback. My email is cvsmith, V as in Victor, cvsmith6405 @gmail.com. Thank you very much.